So I'm Marilyn. I'm one of the graduate students working on the spaceflight Brick 20. I'll be doing the RNA extractions. And this is SVT deintegration. This is the facility where we deintegrated the science. It's where the labs are. This is just walking in to the building. This is our actual lab with Dr. Wyatt figuring out our key code because it changes for high security. This is the patch for Brick 20. Well, this is for generic Brick missions. This is the TV where we could look up our space mission. We're in Lab 456, which was highlighted in yellow. This is Dr. Wyatt getting into our lab. And this is actually getting into the central facilities where the microwave and the autoclave and all of our supplies to do the science are located. This is actually getting into our lab where there was the sticky tape for our shoes to make sure it was a clean area. And this is the 4C where we did the actual deintegration of the science. These are all the people that were important for getting the science deintegrated. These are the Petri dishes that we used. We had different ones that we used at home versus at deintegration. That's Zach, our undergrad, who helped us plate a lot of the seedlings and count them. This is labeling the lids so we know which plates came from which brick because we used two separate bricks. We had one that was a dual chamber and then a single chamber. This is the area where they did the actual deintegration where they pulled out the pens. And that's the frozen brick. That's me taking lots of pictures of the frozen brick. And there it is again. And something cool that we didn't realize was that the brick is actually the size of a household brick. We expected it to be much larger. This is the technician pulling out the pens, and this is an hour and a half after we had let it thaw. You can still see that there's frozen crystals on it, but that's because it was at -80. Those are the tools used for pulling out the plates. That's Dr. Wyatt all geared up for deintegration. And that's the area where after the PDFUs were removed, they were placed so we could take the plates out of the PDFUs. And that's the technician that did the initial removal of the pens from the brick. This is the brick after it's completely thawed and had the PDFUs removed. That was brick A. So this is where we actually removed the plates from the PDFUs, and we had trouble because, like I said, they were at -80, and it took way longer than they had originally thought for them to thaw. So we got this like popsicle effect. We expected just to be able to take the plates out of the PDFUs, but as you can see, the lids were still attached, as well as a nitrex layer, which we eventually, we weren't able to get that out, so that came back home with us. And that's Dr. Wyatt wrapping them. As soon as we were able to get the plates out of the PDFUs and the lids off, we had to quickly wrap them in parafilm and place them into the dry ice without touching the outer rims. That was really important not to contaminate them. So these are the dual chamber. This is brick B. So this is 5 more plates. And again, it was a quick process. As soon as they come out, we had to wrap them in parafilm. And that's all the PDFUs that are deintegrated, so the science is out and in our box ready to take home. And there it is. And all the people there said, "Enjoy your science," which I thought was kind of funny.